Okay, perfect. So, well, thank you everyone for coming to this meeting. Here we have Chelsea. She is a PhD student at the Stacy Smith Lab at the University of Colorado at Boulder. She's working the Physalis genus, especially in evolution, taxonomy, and also nomenclature. She is researching interspecific gene flow, pollination ecology, and reproductive isolation with a focus mainly on the domesticated species and the relatives too. So welcome, Chelsea, and you can start sharing the screen.
Okay. Oh, I haven't shared. Sorry. Okay. All right. Is it sharing?
Not yet.
Okay. I might have hit it too fast. Oh, there's a second button. Is it sharing now?
I can see. Yeah, I can see. Okay. Yes, perfect.
Perfect. As Rocio mentioned, I'm going to be talking about Physalis typification along with some of these preliminary data that I have on self-incompatibility. So first, I want to start with that systematics is important, and I hope to convince you that, and then kind of go over a little more detail of what the work Rocio and I did within Physalis so far. So I think there's three main pillars within systematics. You have your taxonomic work, so your species description and your species delimitation. You have your nomenclature, so this is the work that Rocio and I did. So this is the name, the history, and synonyms, and I'll get into more of what this actually means. And then also evolutionary history, so the phylogenetic relatedness of species to each other. Ideally, groups of species are monophyletic, which means they group together and they have a single group that has a characteristic that you can describe them with. And all of these interact with each other. Once you better understand the evolutionary history, that might change the name of some species, or it could also mean that you figure out that there might be a cryptic species in there. What do you do with species delimitation with that information, and so forth? So the first example I want to give is Physalis acliginki to Acligini officiana. So we probably are familiar with Acligini officiana. It was first described by Linnaeus in 1753. When he described it as Physalis acliginki. With information from Maggie Whitson, we realize that Physalis acliginki doesn't actually fall within the monophyletic group of the North American Physalis or the subgenus Rhyburgius. So this is an issue. So she had a choice to make. What she decided to do is to conserve the name Physalis for all of the New World Physalis. And then also, that means she would have to change the name of Physalis acliginki. So she proposed to conserve the type and the name. And so what this does to the nomenclature is that it changes what formerly used to be known as Physalis acliginki as Acligini officiana. And within this, you can see that there's the history of the authority of who described the species. And then within this, you see a synonym. There's two different types of synonyms. This is called a homotypic synonym. So that means that it's not adjective, and that uses the same type specimens. So I also want to talk about type specimens because these are important. So when a species is described, nowadays you can't publish a species without having a type specimen. This is a specimen that you can refer to so you can look at the description a little bit more in detailed. If it has, so these two will have that same type specimen, so you don't have a different one to look at. I'll get into a little more detail about those differences later on. I think we can learn a lesson from MIMLIS researchers. MIMLIS researchers have been working for a long time on different aspects within this group and family. So we might have heard of Mimulus because it's now an emerging model system, but Mimulus was used with its hybridization and its pollination study. It's been looked at quite a bit for its genetic research, and like I mentioned, it's proposed to be a model organisms a couple times. While all this work was working on in these species, there was also taxonomists and systematics researchers who were working on the other aspect of it. So you can see in this 2007 paper that that these researchers made a point to find these synapomorphies. So these are like the differences within this branch, and to actually figure out what makes these taxa different. In 2012 is when they decided to change the name of certain genera. So this is kind of what happened with the Aclogynki officiana example, is that names need to be changed, they end up changing them. But some of the Mimulus people don't really like the name change. So this is a paper after that systematics work in 2018. They still end up using the old nomenclature. So you can see here that they recognize that there's been a change in the different names and genuses, but however, they choose to use the conventional nomenclature scheme is followed in this review for continuity, the large body of work that previously on ecology and evolution of this group. So what they're saying is that they don't disagree with the work that the taxonomists and systematic people did, but they're just not going to recognize it. I think it's fine to disagree with the taxonomy of it, but we should be mindful with keeping names pretty consistent. What they could do is they could apply to conserve the name, like Maggie Whitson did for Physalis. So I just want to end with really emphasize that systematics is important. It gives us insight on evolutionary history. So instead of just talking about that one genus, they're actually talking about multiple genera in that example on the previous slide. It will give us a better understanding of species diversity, and then helps us with concise genomics. If we really know what species we're talking about, we can pinpoint and look at it. In this era of large data sets, we're getting, there's the pangenome is something that's occurring, and then we're figuring out there's a lot of differences within it. So really having good nomenclature and naming is going to be important for the future. So you really need to know the name of things to study and conserve them. So now I want to talk about the work that Rocio and I did. There's a paper out, an early review. If you want the PDF version of it, you can contact me. I just want to start with a quick overview of Physalis, which I'm sure we're all familiar with. So it's typically yellow and white flowers with dark spots. It's known for its inflated calyx, and some taxa are consumed by humans. The distribution of Physalis is global, with its highest diversity within North America. This paper only looks at species that occur in the United States of America. Like I mentioned, there's a difference between taxonomy and nomenclature. So Janet Sullivan did the taxonomy work for the flora of North America, and we use that as our guide for our work. So there's been a lot of expansions and contractions of the genus. When it was first described, it was 9 different species. It expanded to 120 different species, later to 75 species, and then today it's considered to have 90 different species within the genus Physalis. I also want to emphasize that Physalis is continuing to change, and it's a pretty dynamic group. I wanted to also mention this again is that there's a conserve in the name of Physalis, which is great. And then there's still new species being described. This is an example of that Milo Payne described a new species last year, Physalis macrosperma, that occurs in the southeast. So the objectives of this work was to provide a list of the 28 species of North America and their synonyms, to assess the status of all known type specimens, and to designate lectotypes whenever necessary, and to designate neotypes, which I'll explain what those are, when the original material was lost and there's no illustrations available. The methods that we did is we used online databases as a jumping off point, and then we look through protologs. So protologs are where the species is first described, and monographs, which are a little more in detailed. It's usually has a key, a description of several different species within the group. And then we contact herbarium staff to make sure that those vouchers and those specimens were still at the herbarium. In total, we looked at 28 different species within North America. We looked at different homotypic synonyms. So this is objective. So this is like the example of Aclogynki officinae having the synonym of Physalis aclogynki. And then heterotypic synonyms are subjective. So these will have their own type specimens. So this is taxonomists can disagree of kind of at this stage of thinking that this is considered a synonym, but they might, another taxonomist might actually think that it is a species on its own, or a subspecies, or a different classification, but it should have a type specimen that fits underneath this species. And I'll give some examples of those. I use the International Code of Nomenclature. From now on, I'm going to be referring to it as ICN. Okay, so typifications. What are typifications? Like I mentioned, all types all described species have to have a type. And they, if they don't have a type, then we go back, like somebody has to go back and determine what that voucher is for. And why types are helpful is they're actually really helpful in identification. Because if you have something you collected in the field and you want to you're looking at description, you're doing your measurements from somebody else's description, you might not be quite satisfied with that identification. So what you can do is you can go to the type specimen and compare between those two different things. So it's very important for identification. First I want to talk about first step lectotypification. So this is when a species has been described without a type, a first step lectotypification will happen, and this is just looking at finding original material that was probably looked at by the person who described it. Second and third are very similar. It's what has happened in the past is that people would inadvertently lectotypify before 2001. So this is, they wouldn't mean to be designating a type, but they were when they were writing a monograph or whatnot. And so it's just a couple extra steps to get to that one specimen. An epitype is complementary to a type specimen. So this is if there's not enough material within the type specimen, we'll add an eptype, so it's easier to identify later. The next example is a neotype. Neotypes are, we don't have evidence that the person describing it was actually looking at that specimen. So now I want to kind of go through some examples. So this example, the first example is just a standard lectotype designation. So we were looking at, so this is considered a heterotype synonym because it has its own synonym, or it has its own type specimen. This is Physalis lancifolia, and it's a synonym of Physalis ungulata. So in the original prologue, the original description, these described it without any type designation. So we were able to find one that was housed at MA herbarium, and in the handwriting of Nies. So there's a whole database of handwriting of Old botanists that we refer to figure that out. The next example I want to give is of Physalis hydrifolia. So when Physalis hydrifolia was first described, it was also described with a variety. And it was also with this mixed specimen. What a mixed specimen means is that there's two different species or taxa on the same sheet. So he didn't mention where it was housed. Waterfall inadvertently designated the lectotype of Physalis hydrifolia at GH. And since there was two specimens at GH, we chose the best preserved with the least intermixing. So it's just this left side of the specimen that is the type specimen to refer to. The next example is Physalis finlaria. So in its prologue, it was first described to have two different collections. When it has two different collections, this is called a syntype because it's two different gatherings. And then the first step, lectotypification, was inadvertently done to be this Finlaria 683 specimen without mentioning where it's housed. Waterfall designated it to be GH herbarium. We found both sheets that had a lectotype and an isolectotype that could be evidence of intention of waterfall. But according to the International Code of Nomenclature, the ICN, we can give preference to it, but we don't have to. So we decided to pick the better preserved specimen, which actually has flowers on it to refer to for identification later. The next example is Physalis linkiana, which is also another synonym of Physalis ingulata. So its original description was by Nies, but there was always question about the status. Ryberg quickly made it a synonym of Physalis ingulata. Mahenda considered it excluded or doubtful that was a type, a specimen, because there was no type collection to refer to. She mentioned that there were similarities to ingulata, but the only difference was the length in the peduncle, so that's right, yeah. And she said that it could have been lost in World War II or destroyed in World War II. We searched several different herbarium without any success, but with one exception, we were able to find a specimen with a handwriting of Physalis linka and was able to designate a neotype. So since we don't have any evidence that this was used in the identification or the description when it was first written, it's a neotype. Oh, yeah. One example I want to give is just nomenclature notes. So it's not necessarily anything that has to be done, but just to be aware of, like, this could happen. So the example is Physalis galapagosus, which is a synonym of Physalis cordata. So in the original prologue, it has the collection as 18724, but in his field notebook, you can see the last two numbers are 27, and even on the collection, the last two numbers are 27. According to the ICN, that there's no action that's needed, just to make a note that this typo happened in the original description. Okay, one last example that I want to give is Physalis longifolia. This is another example where there is no type designated. There's two specimens that we found that had the same location, which serve as enough evidence to lectotypify it. But as you can see on this specimen, that it's only leaves and that there are no flowers or fruits. So what we ended up doing is that we have designated this epitype. So an epitype is support for the lectotype that was selected, so it has a fruit and it has flowers that you can refer to, and also the tuber. So all in all, we had 28 species. We have 35 homotypic synonyms, 112 heterotypic synonyms. We ended up doing 53 typifications. 37 of those were first step, 11 were second step, 1 was third step lectotypification. 1 was an epitype, and then 3 neotype typifications. And I would just kind of add this caution that JSTOR isn't always correct with labeling their type specimens. So it would be better to refer to this or other things, but JSTOR is a great jumping off point. Within this paper, we also include excluded names. This is determined by the phylogeny of Physalis by several different groups. In red is strict Physalis, the subgenus Rhyburgius, with the red arrows, with the excluded names that used to be Physalis that have now been changed. With that, talking about the evolution of Physalis, there has been some evolutionary incongruency within Physalis. And so this is kind of the second aspect, or this is my main focus of what my PhD is. So within the different groups, there's a lot of incongruency that happens. So Maggie Whitson first used a 2-gene tree and had low support. Pilar in her group was able to add more species, but the support was still a little iffy. And then when Rocio was doing the whole tribe, she added 3 more species. And while there was some resolution, there is within Physalis, the gene trees were still conflicting and had a lot of incongruency within that. So more sequencing wasn't necessarily better. And there could be a couple different reasons for this. So some might say this is because of rapid radiation, and that causes a lot of incomplete lineage sorting. What that means is that the genes just haven't had enough time to develop unique mutations within their groups and their lineage. But that kind of comes to more conflicting information of understanding if it's rapid radiation, because with molecular dating, the group is only 5 million years old, but with fossil data, it's 52 million years old, and there's several fossils. So this comes to the question, is there potential hybridization and integration of genes? So I think it's almost nice to take even a step back, is understanding the movement of genes within Physalis. So pollen is moved by solitary bees. There's been some studies on honeybees and bumblebees doing pollination. Physalis is considered to be self-incompatible, meaning it doesn't accept its own pollen. And there's mixed results about hybridization. So my work, I've been looking at self-incompatibility in Physalis acutifolia. This is native to the southwest of the US and northern Mexico. It's an annual plant that will persist in warmer climates. It has white flowers with yellow spots. Physalis acutifolia has shown some breakdown in self-incompatibility. Physalis is gametophytic self-incompatible. So what this means is that it is cytotoxic style tissue is created by these sRNases genes. If the pollen tube has like the correct antidote or genetic material, it will deactivate the sRNase genes and grow down to the ovules. So in here you can see a picture. There's pollen grains on top of the stigma. The pollen will grow down the style and then will find the ovule and fertilize the ovule. And some preliminary results have shown lack of sRNase in these self-compatible individuals. So this makes me have another question about is the breakdown of self-incompatibility leading to occasional hybridization? So hybridization in Physalis, like I mentioned, can be somewhat debated. Physalis senescens can hybridize with some species, but not other species. It's thought to be self-incompatible. Physalis, Virginiana, and Hydrophylla can produce hybrids, but are sterile. And in my personal observations, I collected fruits from a Physalis hydrofolia and Physalis heterophylla. and grew out seeds from the hydrofolia to get an intermediate in the greenhouse. So this kind of brings up a common rule with self-compatible and self-incompatible species is called the SISC rule. So if a self-compatible species will accept pollen from other self-compatible species, it will also accept pollen from self-incompatible species. But self-incompatible species will not accept pollen from self-incompatible species and will not accept pollen from self-compatible species. So I've done some preliminary crosses in the greenhouse, and I've seen that my Physalis acutifolia self-compatible line will accept pollen and accept pollen from Physalis philadelphica that was self-compatible. I don't think all Physalis philadelphica are self-compatible. I think I was just kind of lucky to find one that was. And then this Physalis philadelphica, it was not successful with the self-incompatible acutifolia, and the self-incompatible acutifolia would not cross. Physalis cinerescent would not accept pollen from it. Oops. And then the same for Physalis acutifolia so that's what you would expect in that scenario, but the scenario of Physalis acutifolia SI, pollen would not be accepted by this self-compatible Physalis philadelphica, which is not what you would expect in the SISC rule. And then the cinerescence, 50% of the time it was successful between both of those crosses. I think there's several different reasons this could be. It's hypothesized that, well, within Solanum at least, there is some work that has been done that it's genes that are shared in between this self-incompatible system that is shared between this intraspecific relationship between species. And so when sRNase is lost, or these maybe like HT genes in Solanum, that it also loses the ability to recognize pollen from other species. So I think it's going to be a lot more crosses that need to be done, and hopefully unpinning some of this, the genetics of what's going on in there. Cool. And then that's my presentation.
Well, thank you very much, Chelsea.
Yes.
That was great. And yes, and we are open now to questions. If anyone have questions to ask, please, you can turn on your microphone and start asking questions.
I have a question. So everybody can hear me. Oh dear, let's hope this baby doesn't get too loud. Be quiet for a second. So Mike, hold on. Where can I put this thing? So my question is about this situation that's happening in Mimulus, and sort of what's the chance that would happen in Physalis? Because of course you have this weird thing now where, you know, all the papers in Mimulus are going to have that caveat that you mentioned, and all the papers dealing with erythranthy are going to have to mention the Mimulus work. So I wonder if anything like that is going to happen with Physalis. So, and how could we avoid it? That's my question.
I kind of think Maggie kind of helped us, like her foresight to realize to conserve the name was actually really important. I don't know the reasoning behind the Mimulus erythema people, like why they didn't, because it seems like most of the work is done in this American Eurasian group, and that's the one that got changed, because it's also like how nice their phylogeny actually separates by geography. But I don't think Physalis should have as big of a problem.
Can I just and I mean, Joyce will remember this, is that we had a very similar situation with the genus like Persicum and the genus Solanum. And for a while, there was quite a lot of resistance to using the name Solanum for Solanum lycopersicum, which is the cultivated tomato. But the entire community has now switched to using Solanum lycopersicum. And it happened within, what was it, Joyce, like maybe a year and a half or 2 years? It happened very quickly.
It did.
Once
there was certainly resistance.
Yeah, a lot of resistance at the beginning. Well, there was a lot of resistance for about 10 years. Okay. And nobody paid any attention. And so it was partly kind of talking to people, and well, talking to people is always a good thing, but it was very quick, wasn't it, Joyce? I didn't expect it to be that quick. So in fact, it does change. And Mimulus is probably at that slightly awkward time when nobody's really explained it properly, not convinced people, but just kind of laid out the possibilities of what the ramifications of either accepting or not accepting what is a change based on an increase in scientific knowledge means. Because names change for a number of reasons. They change because you find an older name, which is boring and tedious, or they change because we've increased our scientific knowledge, which is cool.
Yes.
So I think explaining that name changes can be quite cool and mean that we've advanced our knowledge is a really important step. And that's what happened with Solanum for sure, Joyce.
Yeah, it was. You could hear the grumblings on people who were like, "Oh, those taxonomists, this changing the name." And but it was. You were right, Sandy. It was a lot quicker adoption than I was surprised.
That's cool.
So we're dealing with some similar stuff with Ipomoea. It's a big nasty genus, and I guess one of the things. We have great molecular data, but it seems that, from where I stand, it's good to be patient until you really have a well-resolved phylogeny. And I think that was one of the issues with Mimulus was that, they didn't have any gene capture data, or, they could have waited for a much more convincing phylogeny. I feel the same way with Ipomoea. We've just had someone suggest a whole bunch of genera be treated as Ipomoea, and I think it's and based partly on a single gene phylogeny. I think that's really premature. So, I think we can propose things, in the literature that tentatively, but to go to changing names, I think one needs to be cautious. That's just my naive opinion.
Yeah, but on the other hand, Rick, when we did the changes in Solanum, lycopersicum and Solanum were done in the 1980s, before we had gene capture technology even. So I think you can make changes based on morphology. DNA isn't the perfect answer to everything. So I think
I agree. I mean, but if you don't even have synapomorphies of any kind, I think if like Ipomoea, find me a synapomorphy, please. We got about 3 in the whole tribe of hundreds of species. So that's where I would say, if you have good. Exactly, molecular data isn't truth, but when you have these really gnarly groups, if we can combine strong molecular data with a careful revisiting of the morphology, but that takes a lot of work, and so I think one needs to be patient and come to that place. And I think when I looked at those Mimulus papers, I thought, that's a little premature. I can see where they're going, but they're kind of hoping that this will be a stable phylogeny with. They have some good, more like she said, they have some good geographic support. But from me, it didn't seem convincing, and I feel the same way with Ipomoea. I think the current monograph that came out was fantastic, but we're a long way from having a convincing phylogeny supported by morphological synapomorphy. So that's just kind of throwing that out there.
Chelsea, can I ask you a question about the self-incompatibility?
Yes.
That's super interesting. And so do you think that this ability to do this leaky incompatibility, which is what Chris Martin would call it, but this kind of leakiness, do you think that's widespread in Physalis, or do you think you found an unusual case?
I don't quite know yet, but I kind of think it's not super unusual. I don't know, because there's some just like really squirrely species like Physalis hydrofolia that's super widespread that I think it's probably hybridizing with a lot of things, and probably heterophylla too. Because I think there's species that have kind of been moved around by Native American people, and like that might have some effect on like growing outside of its quote unquote normal range that might affect like its ability to break down self-incompatibility. Also, I feel like in the Southwest with acutifolia, it's just like the environments that there are the breakdown of self-compatibility are kind of like around agriculture fields or like where there's water all year round instead of like these seasonal
So
like
can I just suggest a paper to read, which would be really interesting for you, is there's a series of papers that were done by Charlie Rick about self-incompatibility in tomatoes, in Solanum pimpinellifolium in particular. And what he found, which I think is really interesting in terms of thinking about ranges of things and where self-compatibility ends up evolving, is what he found is that in Solanum pimpinellifolium, the self-compatible populations were all at the edges of the range.
Mm-hmm.
So always around the edges of the range where it's moving into new territory, things were self-compatible. Yeah. And that's true in a lot of species of Solanum, so it might be true for Physalis as well.
Yeah, which is cool. Physalis also has like an interesting history with its self-incompatibility, just because it's thought that like Physalis has gone through this whole bottlenecking event and then had a rediversification, and because of this rediversification, it has a lot of different S alleles than Solanum, which I'm also interested to see like how that might affect the genetic diversity as a whole, maybe later on, of like the population is self-incompatible might not be as bad for these species just because they don't have as many deleterious mutations within it. Okay.
Okay. Here we have a question of Steve about the typifications. And he asked when did the typification started? If it is in carb only networks, there is no type designation, which is right. And species plantarum, he just gives a list of species for each genus, but no type designation.
Actually,
yeah.
don't.
So actually, Sandy's asking, answering that because the types only have been, sorry. Go ahead.
The type concept is very, very new. So most of the botanical literature existed before we had a concept of types. And so according to the code, types only have to be designated for names that are published after 1953. So anything before that, or 1935, I can never remember which it is. I'm supposed to be an expert, but I don't know. So it's actually in the 20th century that the type method became sort of fixed. Now, people were thinking about types and saying that things were types. So the idea that there was a physical object that you could tie a name back to is something that came from de Candolle, who was a Swiss botanist in the 19th century. So the idea has been around, but it's been the rule only since about the middle of the 20th century. So a lot of nomenclature, a lot of doing monographic work is actually the really hard slog of figuring out what people thought in 1852, and which can be impossible, and you kind of have to be quite practical and creative sometimes. Does that help?
Yeah, the explanation was great. Actually, yeah.
I didn't realize it was.
I can say one thing about epitypes. Can I just say one thing about epitypes? Is there's a trend amongst people at the minute to designate epitypes for anything that doesn't have molecular data, so people can have molecular data. You can only designate an epitype if there's substantial difficulty in using the type, the type specimen. So it's not something that you just do just because you might want to have something to extract DNA from. Or something that has molecular data. An epitype is to be used very sparingly and only for places where the specimen, like Chelsea showed, doesn't have the diagnostic features. So let's say the diagnostic feature of a species of Physalis is what the inflated calyx look like, but the specimen is only in flower. That's a good case for having an epitype. But people are starting to kind of designate them willy-nilly or whenever they like, which is actually very, very bad practice.
It's good to know.
And there is also like another question of Thibaut about that he knows that it's mandatory in the Code of Nomenclature since 1953, but it's in the lectures before. That was the question. When did it start actually? So the Candole, yeah, right. When was the Candole?
Wait a minute. Sorry, my microphone was muted. So de Candolle wrote a paper called Les lois de la nomenclature botanique. If you would like to look into the history of the code, if you'd like to look into the history of the code on the IAPT Global website, there is this guy has done this amazing digitization of every single version of the International Code of Nomenclature for what is now called algae, fungi, and plants, but used to be called botanical nomenclature. And you can then trace back all of the different articles back to the very beginning. But de Candolle is the person who first started talking about types. Interestingly, he didn't use them. So in the Great Prodromus, which he wrote, which he and his son, Augustin Pyramus and Alphonse de Candolle, 2 de Candolles, not just one, there were actually 3 botanical de Candolles, thought about that, but then never used the words type in their own work, which is the Prodromus. So it's something that people had thought about, and Lamarck thought about it as well in the 18th century, but people didn't use it as a concept.
That's a great explanation. So yeah, I don't know if Thibaut, you have another question about that? He's typing. Thank you very much. Thank you, Sandy. Any other question about self-incompatibility or the types? I don't know if Craig, Pilar, Franco, someone has a question, or Anna, that he is also working in some nomenclature for the convolvulaceae, actually.
I have more of a point for discussion, which is like, so let's say we find out that Physalis is 53 million years old. That's you know, that holds after Rocio's work. I wonder about, you know, that for animal people, hybridizing after 53 million years separation would be pretty shocking. Although maybe they've kept hybridizing the whole time and sort of held on to that possibility. And I wonder, you know, there was a paper by Carl Rothfels that I remember made it to like NPR, and it was about fern species still hybridizing after, I think it was 60 million years. So I wonder how many other groups we have that kind of information about. They're still hybridizing, they're still able to hybridize, even though maybe they were separated for a super long period of time. And I don't know in Physalis how many of these species have historically hybridized, possibly, you know, this entire time, versus, you know, they've been separated, some in North America and some in South America, and you bring them back together and they can still hybridize even though they've been apart. So I don't know, Chelsea, how many of the species, I know you have Peruviana in your mix, and you've been doing crosses with that. And I don't remember your results, how easily that can cross with the North American ones. Anyway, this is more of a point for discussion, not a very well-formed question.
I haven't done too much actually with Peruviana, just because I know it's not diploid and it's, yeah, so it's self-compatible, which is nice, but it's such a big plant. So I don't know.
Well, when we worked on Nicotiana, which are actually, depending on how you look at it, they're probably a slightly kind of older lineage than Physalis, is that after about 10 million years, the alloploid hybrids you can no longer distinguish the parental genomes using fish, and so they become completely diploidized and they don't look as though they're of hybrid origin. So there's lots of kind of hybridity that's kind of not maybe seen as hybridity. There's also, I think, a problem with kind of imputing hybridization to kind of answer what, Chelsea, you perfectly described as gnarly species, you know, species that have loads and loads of variation and are really complicated. Because that happened in potatoes, so potatoes would be, people describe species of potatoes, and then they would say all the variation in between is just due to hybridization. So it was almost a way of kind of pushing pushing variation to one side rather than trying to show how variation occurred and why variation was occurring, because variation is a biological thing. So hybridization can be used as I mean, I'm not saying that you're doing it, Chelsea, but people have used hybridization as a way of ignoring variation, trying to push variation to the side.
Hi. I just have a comment about the hybridization in Physalis. We have been in the field and with species that are species that in cultivation, around the corn cultivation in the milpa, we have found even 11 species of Physalis, and they don't show hybridization. That is a very, very important thing to comment. And however, we have observed Physalis philadelphica in the greenhouse being self-incompatibility. So maybe there is a thing of lucky, I don't know, to find these plants with to find these plants who are tough. There are I'm sorry,
it's okay.
A little nervous about my English. Maybe...
Don't worry, you can speak in Spanish if you want. Fine.
Ok, va. Entonces, bueno, por favor me traducen. Lo que nosotros hemos visto en cultivos donde hay, alrededor del cultivo de la milpa hasta 11 especies de Physalis que no se muestra que son híbridos, ¿no? O que no pareciera que no forman híbridos. También hemos tenido en jardines ahí en nuestra escuela, en nuestro centro universitario, donde no vemos este proceso de hibridación, ¿no? Ni formas intermedias. Donde lo hemos observado más bien es en como lo que llamamos nosotros complejos de especies. Por ejemplo, Physalis crassifolia, la acabamos de ver en toda la Baja California y parece ser que en ese complejo de especies, porque tiene muchas variedades, hay como este proceso de hibridación, pero yo no sé qué tanto entre especies. Me parece como, raro. A nosotros nos parece raro ese proceso.
Translate for everyone. So she's saying that usually they don't see these hybrids in the different species of Physalis. Actually, they have the sample 11 species. They haven't found that. But for some complex of species like Physalis acutifolia, they have seen some hybrids that are only between varieties, which means that there are not very clear limits about those varieties and to be just a complex within that species of crassifolia. I know if that's clear, Pilar.
So you're kind of breaking up. There's a lot of variety within species, but what was about crassifolia that there has been?
Oh, that within crassifolia there are varieties. So yes, varieties. Sorry, and there are kind of intermediate morphological plants for them, but it could be just because it's a complex, and actually because there are not different species. So there's all are within the crassifolia complex. That's very interesting because actually, so you haven't seen in Mexico or in doing among these sample species hybridization. That's very interesting, because we have seen something that could be intermediate potential hybrid in, which is this park, Chelsea, I can remember, that we saw the Pterophylla and Acutifolia intermediate.
And Pumila and Hispida kind of, or was it like Pumila and Virginiana? I think there's some that are just kind of squirrely and might hybridize with others.
That one, right. Yeah, it's the Lorry State Park. Now I remember, Stacy. So.
Yeah, that one. But it also, like looking at herbarium specimens, I think we saw one that looked kind of, it could just be variation, but it looked like Physalis Pumila was mixed with Virginiana, which that's been documented by Hinton, the Virginiana heterophylla hybridization before.
So, yeah, and actually between Pumila and Hispida has been considered, I think, a subspecies of Pumila. So it makes sense if there is hybridization between those two species, because the limits are very fuzzy.
Can I follow up on what Stacy was talking about a little bit? Which is like, you know, when we talk about syngamians and plants, you know, flowering plants, which are really common, I've always kind of, I don't know whether it's a shared assumption or not, that these are temporary states, you know, and that eventually these species will all sort themselves out into nice little packages. But, you know, I've been starting to think, you know, how long do these kind of how long do these arrangements last? You know, like you're saying, do they last for millions of years, or are they just something temporary? You know, this sort of the gray zone of I don't know his name, how to pronounce his name, but Decorzo or whatever. You know, sort of that the species, that populations will move through this gray zone, and then they'll all be good species someday. And I guess I'm starting to really question that.
Yeah, I think that's a hot debate, and I think it's like, where are you on the evolutionary trajectory? Like, is it a trajectory? Not really. Like, it's just stochasticity all at the same time. Yeah, but how long things stay in one state, I don't know. Maybe somebody else has an opinion about that.
I don't know either, but you know, this is always one of my beefs, is that it seems like we always see the line towards speciation as a straight, you know, one-way street, but there's no reason that things can't diverge for a bit and then fuse, and understanding, you know, where we are at any moment in time, I think is super, super hard. And it's definitely dangerous to assume that things are on the way to speciation. They may be on the way to not speciation.
Mm-hmm.
Yeah, I think there's something else that Joyce, before she left, she said that she has seen in Physalis peruviana from the growers in Colombia that there are tetraploid, diploid, and mixoploid types of peruviana. So that's very interesting. Like, I think we have seen that with Franco too, that there are multiple different samples of the same species can have different chromosome numbers. We have seen also with one myochroma. I can't remember the species, but Franco might remember better than me. But yes, that's pretty common within Physalis, I think. Franco, do you remember the name of that iochroma, or probably Stacy also remember?
I thought it was Fuchsioides, but I don't remember. But I think it's definitely adds another level of possible complexity, because if species have multiple ploidy levels, then maybe, you know, the diploid populations can cross with other diploids, the tetraploid population can cross with other tetraploids. And I think we just don't know. We don't have population level data for hardly any species. So I think we have a sense of how much within species variation and ploidy there is for any of these taxa. But like I said, Franco and probably know better.
Yeah. But Franco, I don't know, probably his microphone is not working. Okay, any other question, or yeah?
Looks like some coming into the chat box, Rocio. I don't know if Maria.
Oh, that's true. Yes, Maria and Rick are typing, so we'll wait Oh, it's just amazing presentation for Chelsea. Let's see if Maria.
So next presentation in 2 weeks will be Joyce, and yeah, I think she's going to talk about some of the work she's been doing with the cultivation of Physalis peruviana and Griesii and or Grisia. And I don't remember if there's another species.
Grisia and peruviana, and I think Philadelphia too.
Nice. Cool.
Okay. So, well, thank you very much, everyone, for coming. See you in 2 weeks.
Yes.
The time will change here in the London, so I will update that timetable, but it will be the same for the US folks and all the other parts of the world, I think.
Okay, sounds good. All right.
Okay. Bye. See you.